Well, thank you very much for the opportunity to talk to you about this interesting project. And I'm going to demonstrate my Canadian roots by starting off this talk by apologizing to anybody that was here earlier in the day and saw Dan Ewell's excellent talk, that you're going to see some slides that are, again, so if you have a fuzzy feeling of deja vu, that's because you vooed it deja. So the second thing is to note the cast of characters involved in this project, again similar to what you saw this morning, and just to thank this group of individuals, in particular Dan, who's really led this project in a big way. It's difficult enough to do stock assessment and morphometrics on one of our Great Lakes, let alone 5, in somewhat of a timely fashion. So this is a really good example of cooperation to produce a pretty interesting data set. So the overarching goal of this project is eventually to establish a database of morphometric and genetic data on the same fish from the genus Coregonus. And just as a reminder, even though I'm not going to talk about any of these species today, the other species that we're working on right now, at least the extant forms of the deepwater Cisco group, and the hope is also to try and also generate or to get some historical data into this database as well. So as I said today, the rest of the talk is going to focus on the Artidae, the Coregonus Artidae, the Cisco or the lake herring in the Great Lakes. They, as you probably have heard once or twice or more, they are historically and also today important in fisheries and food web in the Great Lakes. They have declined as a result of overfishing, invasive species, habitat loss. So these large catches that used to be seen once upon a time have been greatly reduced. In Lake Superior, the fishery is holding steady to maybe declining, as you saw earlier in the week. And in the other lakes, they're either extirpated or greatly reduced. But as you well know, there is possibly an opportunity for rehabilitation, and this is I guess a fairly hot topic in the discussions of many lake committees across the Great Lakes. So some of this work is coming at a pretty good time. So the general questions that we're asking in this part of the project is, first, how is genetic diversity of Coregonus partitioned across the Great Lakes? And I'm going to talk mostly about contemporary data today, but as I said, we are interested in getting a handle on what was out there historically as well. Second, and this is the part that Dan talked quite a bit about this morning, was do the historic forms of the Coregonus Artidae or the Cisco persist, and if so, where are they? And then the third part that Dan alluded to this morning was, is there a correspondence between genetic and morphological data? And then finally, given all this information, what are the best possible sources for rehabilitation if a stocking program is the way people tend to go? And I'm not going to really head-on address this question today, but just so you know that it was apparently something that people are interested in. So we are starting to think about it, and in true fashion at the end, I'll probably pose more questions than answers related to this question. Anything I do talk about, it'll probably be a really fuzzy discussion at a fairly large scale. So just a really brief overview that back in 1929, Walter Kautz described several forms of Cisco in the Great Lakes, and the two predominant forms were the Coregonus Artidae and the Coregonus, or at the time Leuciichthyes, Artidae albus. Now there was again a third form, but for most of the rest of the talk I'm going to be focusing on examples morphologically at least of these two forms collected from areas where they were historically present. So just as a review, these two forms, the Artidae was the slimmer form with the longer head, and then the albus was like the deeper bodied form with the shorter head. So historically, you saw the Artidae form was most predominant in all the lakes except for Lake Erie, where you did see that the albus form was the predominant form. So the sampling area that we covered with this project was basically all across the Great Lakes, and today I'm going to talk about fish from all of these sites with the exception of the Manitoulin site. And also Dan earlier did talk about some fish from Lake Erie. I'm also going to talk about a collection of fish from Lake Erie, but they're not the same fish that Dan talked about. He talked about some contemporary fish that some fish that have been collected in commercial catches over the last few years. I'm actually going to talk about a sample of fish collected in 1927 and 1928 during from a commercial fishery. We used scale samples to try and get some genetic information. So in all of these cases, including the archival samples, these fish were all collected during the fall spawning period, and there is, with the exception of the older samples, there's a minimum of about the average sample size was about 50 fish per collection. So it's a pretty healthy sample size from across all the Great Lakes. So we used a pretty standard approach for population analysis. We looked at microsatellite DNA data using 11 loci that are designed, if not for Cisco, but then also or alternatively for whitefish. And we looked at genetic differentiation among the sites that I just showed you on the previous map using a couple of approaches. First, we used an individual-based analysis using a Bayesian approach that basically took the individual fish without considering where they came from to try and generate the most likely number of populations based on the genetic data that we had. And then second, we used several cluster-based metrics that are basically you take the sample sizes, the sample collection sites that you have, and then you look at differences between those sites using a variety of metrics. And then the idea is that there would hopefully be some kind of correspondence between these two approaches that would give you insight into the population structure. And then the final thing that we looked at was the contributions of the collections that we made to the overall genetic diversity that we observed across the Great Lakes at the end in the full data set. So just before I jump into some results, I'm just going to show you a couple of examples of the type of results that you see on these individual-based approaches, just so you can get a handle on what the different kind of results can mean, because you see these more and more often these days when people talk about genetic analyses. So with the individual Bayesian-based approaches, at the end of the day, you run all your samples, you figure out what is the most likely number of populations, and then what you can do is you can look at the individuals and say, well, for each individual, what proportion of that individual's genotype arises from from each of the populations that you've sort of estimated. So on one hand, if in this example, it's a hypothetical set of data where, say, the most likely number of populations was 3, what you could get is for each of the bars, vertical bars represents an individual fish, and the different colors represent the proportion of that fish's genotype that is associated with each of the populations. So in this case, what you have here is a set of 3 populations that are pretty well differentiated, so that each individual fish, the majority of its genotype is associated with a single population. So on the other end of that spectrum, again, here's another set of hypothetical data where, again, the population size that you get is 3, but in this case, it's not as clear-cut, which might indicate that these populations aren't quite as divergent as in the previous example. So with that in mind, we'll move to the results that we got from a data set. So again, each of these individual vertical bars represent each of the 760 plus fish that we looked at. The fish are arranged sort of roughly west to east in terms of their geographic origins. So what we got was an ultimate sort of the best estimate was somewhere between 3 or 4, depending on how you looked at the analysis, was the number of populations that you found. And when you put on the sources or the origins of the fish onto the graph, you can see that that in general that you start to see a pattern where the fish from Superior are fairly distinct from the fish from the other sites that we collected. So there is some structure between at the basin-wide scale. So I was kind of, to sort of look into this a bit further to see if maybe the large number of Superior fish weren't obscuring some of the structure either within Superior or within the other lakes. We broke this data set into 2 and ran the analysis again. Now, when you just looked at the Superior fish on their own, you didn't really see any you basically saw 1 to 2 populations when you broke that apart. And when you looked at the samples from the other lakes on their own, you did indeed find that there was a little bit more structure that the full data set was obscuring a little bit. In this case, there was again 3 to 4 populations is what we were seeing, and in particular, you did see that the fish from Ontario and the Michigan, the Grand Traverse Bay sample were quite distinct. So to move on to the results from the cluster-based analysis, This is the same diagram Dan showed you this morning, where each individual sample genetic distance metrics were calculated for between each pair of samples and then visualized on this genetic tree. The best support that we saw were for the branches from the Grand Traverse Bay samples, which are, again, quite distinct, as with the individual-based analysis, and also for the samples from Lake Ontario, which were quite distinct. And again, you can see that the samples from Lake Superior are in a group together on the top of the diagram. So at this point, we were able to get data from the samples that I mentioned about in the beginning from Lake Erie, that we were able to get a subset of the loci. We were able to get data for these fish, so we've added them into, at this point in the analysis, just to see where they cluster out. So there were 2 samples, one from Sandusky, a port on the south shore of Lake Erie, and then also a sample from Port Dover in the Ontario waters of Lake Erie. And again, although this branch was not particularly well supported, it does show that these samples from Lake Erie were quite distinct. And we are investigating this a little bit further, trying to collect additional samples, add them to analysis, and improve the number of loci that we can bring to this analysis. So then the second part of the cluster-based analysis that we did was to run a couple of the metrics that looked at on the top half of this. This chart is basically an upper and lower triangular matrix of some values, and don't give yourself eye strain by squinting at the actual values. But what we have here is for each collection site, we could do a comparison in one of 2 ways. First, we could look at, is there a difference in the allele frequencies between each set of collection sites that we have? And then the second thing was we could look at a standardized measure of diversity to see if a allele frequency variance was different between each of these sites. So what we have on the top half of this graph, and these are organized, they're color-coded by the source, so all the Superior samples are one color, the Michigan sample is in another color, and then the other 2 lakes are in another 2 other colors, so you can just sort of see them by lake. So the top half of the graph is a probability of whether or not these samples are, the allele frequency differences are probability of the differences being the allele frequencies not being different. Those are on the top half of the graph. And on the bottom half of the graph is the allele frequency variance, which is a scaled estimate from 0 being that there is no variance between allele frequencies in a set of collections, meaning that they possibly could come from the same population, to a maximum of 1, in which case these are totally quite, extremely different. So looking at the probability values, everything that's blocked in here, this is an incident where the allele frequencies are probably not different. So they potentially could belong to the same population. And on the same hand, the ones that are just blocked in are where the allele frequency variances are not significantly different from 0. So these potentially come from a similar population. So there is not an overall correspondence between these 2 approaches, but when you sort of look at it overall, you can see where there, for the most part, where there aren't differences, again, as with you've seen in the previous 2 sort of types of analyses I've shown you, this generally corresponds to the Superior versus everything else. So the samples that we got from Lake Superior tend to cluster very closely together. And these, I'm going to jump back a little bit here. This one right here. And this result, you actually have seen something similar like this before in a previous analysis that Kim Scribner did on some Cisco from the Upper Great Lakes. And also, if you kind of remember, if you happen to remember a few years back, there was a study done on some lake whitefish from the Upper Great Lakes out of Brian Sloss's lab. It was Justin Van de Hee's master's work, that they also found that some of the samples from Grand Traverse Bay were quite distinct from the rest of the samples in their collection. So this kind of pattern has been seen for Carignan species before. And I'm just going to jump ahead. So the last thing I wanted to talk about is the overall contribution to diversity. So the way this graph sort of is laid out is that you can look at diversity in sort of 2 parts. First, you can look at, for each of the collections, you can look at their contribution to total diversity based on the sample's diversity. And the second part, you can look at on their contribution to the overall diversity due to the divergence of the population from the average of all the populations. So a sample that's quite distinct genetically would probably contribute more to the total diversity than one that isn't as quite distinct. So the other thing to note about this scale, even though you're seeing some pretty differences in one, or pretty large differences in one of the samples in particular, you do have to note that the scale on this is a percentage, and the largest numbers are 1. So overall, even though the Grand Traverse Bay does contribute more than average to the population diversity, and then a little bit less than average to the sample's diversity, it is still at most 1%, which is consistent with the small amount of divergence that we are seeing. So on this next graph, this is the comparison of the morphometrics to the genetics that Dan talked a little bit more, that we're in the process of working on right now. So this, on the left side of the graph, is basically the morphometrics tree diagrams that Dan showed you this morning, which shows that shows how the samples cluster, the different collections cluster based on their morphology. And on the right half of the graph is the genetic tree that I showed you a few slides ago, just represented in a different way so this comparison can be made. So we're trying a few different analytical approaches to get at this with the help of Gene Adams. And this first diagram that I'm showing you is called an entanglement diagram. And the theory from it actually is derived from quantum physics, where it's a type of approach to an analysis that you use when you want to look at characteristics of particles that have become closely associated, so that if you want to look at sort of the characters of one particle, the only way to do that is to also have information on a second particle. So what it means then is you get a measure as to how entwined or combined are two sets of information. So in this particular graph, this entanglement score is fairly low. And then we also looked at a couple other more traditional similarity metrics, looking at the Baker's Gamma Index, which was significant, saying that these two trees are similar. And then the other thing we looked at were a cophonetic correlation measure, which actually indicated that they were not correlated. So overall, even though these two data sets that we have, they are similar, but they are not correlated. So what does that mean? That's a pretty good question. We're going to be looking into this particular aspect of the data set a little bit further, and trying a few other approaches and analyses in the future. So stay tuned for that. So to summarize, the first question was, how is genetic diversity partitioned across the Great Lakes? What we are seeing are small but significant differences across the Great Lakes. Particularly at the lake level, the lakes are most distinct. Within Superior, it is possible that there is some structure within the lake. Huron is quite distinct. Michigan is particularly distinct from the other samples. Ontario is distinct from the Upper Great Lakes, and you did see some, the two samples within Lake Ontario were genetically different. Now, even though we are seeing this on the basin, most of the diversity we're seeing is being driven at the basin-wide level, you also have to remember that for 2 of the 4 lakes, we only have 1 sample. So that could be as much a result of the sample design as anything else. And we are sort of trying to acquire a few more samples from Lake Huron and Lake Michigan. But, the take-home measurement is there is diversity, but it's small but significant structure across the Great Lakes is kind of the take-home message. So overall, if you look at sort of genetic diversity across the Great Lakes, there is a similar levels of diversity overall, with the exception of Grand Traverse Bay, which contributes more to the overall diversity because it is so genetic. It is genetically distinct. Just sort of put this on a graph. You can see that the bold sort of circles indicate sort of major breaks in genetic divergence, and then either secondary or more subtle breaks in divergence are indicated by the dotted lines. And just as a reminder, the circle on Lake Erie represents the historical collections. These are not the Ciscos that Dan was talking about this morning. So then, just to sort of remind everybody who was here, and if you weren't here, to answer this next question, do historic forms of ardei persist, and if so, where are they? Just one slide to summarize what Dan talked about this morning was that in Lake Superior, you are still seeing today the ardei form as the predominant form. In Lake Huron, you he did not, you are seeing the Albus form today as the predominant form. In Lake Michigan, as in the past, the Artidae was the predominant form they've seen in the collections they've analyzed to date. Interestingly, in Erie and Lake Ontario, you do notice that on the contemporary samples that Dan has analyzed, that they are being classified as the as the Artidae form, whereas in the past it was the Albus form that was described as the predominant form. And in Lake Ontario, again, you are seeing today the Artidae and some of the Albus forms. So there has been, in Lake Erie in particular, there has been sort of an interesting shift that we'd like to investigate further. So this final question, and also, do morphometric differences correspond to the genetic differences? So we saw a high similarity between the two dendrograms, but the overall correlation is weak. And as I said, we are working on other methods of the analysis to look at this further. And then, so the final question, so what would be the best sources for rehabilitation? So that is a good question. There are some information gaps. The data that we're showing today is one piece of information. It's one variable in a pretty large equation that needs to be figured out if you are going to be looking at stocking as a method of rehabilitation. This is one piece of information that you need, and there are others. Some of this information is available, some of it isn't. In particular, we'd like to expand on the data that we have to get a more thorough comparison of historical and contemporary data. And really, really important, it's important in sort of any rehabilitation program that involves stocking, but particularly important, I believe, in corygonids, where past research by Tom Todd has shown that their early rearing environment can have a huge impact on the morphology of this critter, more so than many other species in the Great Lakes. So it is really important to understand the role of genetics and environment in morphological and life history variation, which is something that Dan discussed also this morning. And to this end, I'm involved in a couple of projects that will be investigating using next generation bioinformatics approaches to improve the sensitivity of the genetic markers that we have used to date to look at population structure, and also possibly to sort of starting to get to the question of what are the relative roles of genetics and environment in the establishment of these different forms. So this is an important line of research that I think needs to be done to start to answer the overarching question. The other thing that is, if stocking is the way to go, it's going to be also important to have more data on the potential donor sites to add to this database so we can do some comparisons. So